defined as all of the chemical reactions that occur. All chemical reactions that occur in living systems, that's metabolism. But you can really split it into two categories. It's either the synthesis reactions or the decomposition reactions. So we're either building things up or we're breaking things down. Anabolism is which one? Anabolism are the building. I remember it because I always think of anabolic steroids. So people take anabolic steroids to like build muscle mass. So anabolism and the anabolic, it helps me to remember the two. Critical things with anabolism compared to catabolism, it's really like the opposite. Catabolism is breaking down molecules. Where you take bigger molecules, you break them down into many small molecules. So remember decomposition reactions? You start off with something that's big and you end up a lot of little pieces. Anabolism is like a synthesis reaction. Anabolism is taking little building blocks and then building large things. So you see that they're sort of the reverse of each other. So this is your decomposition reactions, whereas these are synthesis reactions. Which one makes energy? Which one releases chemical energy? Anabolism or catabolism? What do you think? Do you think that you release energy when you build molecules, or do you release energy when you break things down? Break them down. That is the whole point of catabolism. This is the energy producing part. And it's what powers the synthesis reactions. So we can break down molecules and release the energy contained within the chemical bonds, and then we can use that energy to be able to build molecules that are needed within the cell. So this is, one is the energy producing, one is the energy demanding. And so this little sort of flow is a good example of how anabolism and catabolism are like used one to the other. Take small building blocks, you can use them to build. There's your energy demanding reactions. Take the big molecules that come in and you can break them down in order to generate that energy. So the energy is called ATP. It's adenosine triphosphate. Kind of looked at its structure before. And it's called the universal energy molecule of the cell because all energy gets converted to ATP. All chemical energy gets converted to ATP, and it's a uniform energy source. So every time you use ATP, you get the same amount of energy. So it's a nice, even, constant, and storable energy source. So it's a uniform energy unit. Some chemical bonds have a lot of energy in them. Some chemical bonds don't have very much energy in them. And so this, by putting everything into the same energy unit, it's going to give you constant and even energy, and it's somewhat storable. So you can store it in the cell. If you don't need it, it'll just stay there kind of like a battery. It's still going to hold that charge. It's still going to have the energy until you actually need it. So you can store it as potential energy. And it's what you use in order to be able to build molecules in the body. So when thinking about the process, and I know I got these ones out of order. So when thinking about the breakdown of molecules, so here's the first one. So I did carbohydrates. So the first example I've got is the simple sugars. So in here, find glucose, fructose, and galactose. So on this chart, do you see glucose, fructose, and galactose? Do you notice that they're the smallest? Okay, so here's galactose, here's glucose, here's a glucose, Here's fructose. Those are the monosaccharides. So they're the single little sugars. They don't get digested at all, okay? So the monosaccharides, there is no digestion. They get absorbed as is. So glucose, fructose, and galactose, they don't require any digestion. They get absorbed. In fact, glucose can get absorbed directly through the lining in the mouth. So absorbed through the mucosa lining. Other monosaccharides, as they're produced, they're going to get absorbed quickly. So remember, we talked about them and how it ends up affecting blood sugar, how because there's no processing, there's no slowdown in digestion. As soon as they can be absorbed, they're going to get absorbed into the bloodstream. So monosaccharides require no chemical digestion. So the disaccharides. So the disaccharides, finding those, maltose, lactose, sucrose. Notice there's no processing of these until you get down into the small intestine. How much digestion do they have to have? You've got to take maltose and do what? One step. You take maltose, you split it into two glucoses. You take lactose, you split it into a galactose and a glucose. So you only have to break one bond. So is that going to take very long with enzymes? Enzymes like maltase, lactase, and sucrase? No. Remember, enzymes speed chemical reactions up thousands, sometimes millions of times faster. So this is really pretty rapid. Not as fast as the monosaccharides, but still pretty quick. So they're absorbed pretty early in the small intestine. They're not going to have to go through that entire 20 feet of the small intestine. Typically within the first couple of feet, they're already digested and absorbed. So they'll affect your blood sugar, not quite as fast as the monosaccharides, because you do have to split them in half. But now what about the polysaccharides? And I wish they, tried to actually illustrate it a little better. So here's their polysaccharides, and they really only put seven glucoses in the link. But in fact, remember that polysaccharides are hundreds or thousands of glucoses. So it's a whole lot more. So in order to be absorbed, what do you have to do? You got to break them all the way down to single glucose. So you're talking about breaking hundreds to thousands of glycosidic bonds. You've got to break the links between every single glucose. You can't absorb pieces of polysaccharides. You have to get all the way down to the individual units. So we're going to have to break hundreds to thousands of bonds. In order to get all the way down to the glucose size. So these are the ones that are going to take time. These are the ones that are going to get halfway, 2/3 of the way through the small intestine before you really start absorbing glucose. This is why you have slower changes in blood sugar when you eat polysaccharides compared to those monosaccharides, because there's a whole lot more processing. So it's a longer digestion process, but eventually they will all end up as glucose. Polysaccharides are all made of glucose when you're talking about starches. What about cellulose? I did list that one in there. What's unique about cellulose? You don't digest it. So it adds bulk. It helps to slow the absorption of some of the fats and sugars as it passes through small intestine. Remember, fiber has some beneficial characteristics, but it doesn't fall into the starchy polysaccharide category. Okay, so then what about lipids? So what's the biggest challenge with absorption and digestion of a lipid? When you mix lipids with water, what happens? They separate, right? Oil and water, fat and water, they're going to separate out. And so in order to keep that from happening, so lipids They don't mix with water. And so you use bile. Bile is just like soap. It's made by our liver. And it acts like soap. So what does soap do? Soap forms those little micelles. And so anything oily gets pulled into the micelle, and so that's what happens. So the fat, the oily part can get pulled into the micelle of the bile, and that allows enzymes to break your fats and oils down into smaller fatty acids. So most lipids exist as what they call triglycerides. Triglycerides is just 3 fatty acid chains linked together. So that's the tri. 3 long chains of carbon just linked together. And so you're going to break that down, and the bile helps that all to interact. So it's going to help the fat and the oil to mix with the watery environment that's in the small intestine, allowing the enzyme to come in contact with the fatty material, and that's going to help to break it down. So the goal kind of is still going to be kind of oily, non-polar, not going to mix with water, but as long as that bile's there, it's going to keep it from separating out. You don't want it to separate out, because if it separates out, it's not going to get absorbed. It's just going to end up passing all the way through, and that's a holy mess. Okay, so they call that emulsification. So it is going to emulsify. All that means is it's just going to help them to mix. It acts like, just like soap helps your water and dirt and grease to mix. Same thing with bile. It helps fats and oils to mix with water. That's going to allow it to be absorbed. Once fatty acids get absorbed, bile, some of the bile goes with it, not very much, but a little bit. In fact, that's what gets filtered out in your kidneys as a urichrome is what it's called, and it gives urine like a yellowish color. It's actually a bile pigment. So sometimes some of the bile gets absorbed, most of it doesn't, but you have to have a carrier even in the blood. So one of them we talked about. What was that called? Nope. What were the carriers for fats and oils? The cholesterol carrier is called, there's 2 abbreviations. The HDL and the LDL. Do you remember those? So those actually carry cholesterol, but they also carry fats and oils too. Because even in the blood, fats and oils are just like oil and water. They're going to separate out. So they actually get transferred from bile to another blood carrier to be able to be transported. So they can get sent to cells to be used for energy, or they can be broken, or they can be stored in adipose tissue because they can be stored without water. So they do have carriers in the blood. The initial carriers, these little chylomicrons, are kind of like rudimentary HDL, LDL, and then they get further processed, and that's how they're always transported. When they talk about triglyceride numbers, that's sort of your initial transporters of fats and oils. So if you ever get your blood test, like just get a regular blood panel, they talk about HDL, they talk about LDL, and they'll also talk about triglyceride numbers. So that's just looking at these initial sort of circulating fat molecules. Proteins, the last one, I skipped it because I didn't list it first. So proteins are the chains of amino acids, and you can really think of an amino acid as roughly the size of a glucose. So proteins tend to be anywhere from 25, they can be a couple of hundred of amino acids long, so they're not as big as polysaccharides, but they're still too big to be absorbed directly. So they have to get broken down. So remember that these are amino acids. There's enzymes that are present in the stomach. Hydrochloric acid, stomach acid, helps to speed up the processing. But you have to do hydrolysis, break the peptide bond, just like you have to do with the sugars in the polysaccharides. break them all the way down to their single units. So they can't get absorbed as an entire protein. You have to take that protein, process it all the way down to the single unit size in order to be able to absorb it. Starts in the stomach, finishes up in the small intestine. So that means that after digestion, so if you have a mix of carbohydrates, proteins, and lipids at the end of digestion into the bloodstream, you are going to have glucose, because that's what all of the carbohydrates end up going as. Jeez. So glucose goes into the bloodstream. This pool of amino acids, remember we said that as long as you have those 8 essential, the other ones can get made, but you're going to get amino acids. And then the fatty acids, which are the little lipids. Carbohydrate, protein, and fat oil digestion. So you get all 3 of these, and now they're circulating in the blood. You can use any of these for energy, which is really what the whole second half of the chapter talks about. We can break down glucose for energy. We can break down amino acids for energy. We can break down fats, fatty acids, long carbon chains for energy. So you can use all 3 of these. But in order to break them down, there's stepwise processes that we want to do. So the first thing is they talk about some of these energy intermediates. This sort of shows like the general flow. Amino acids coming into the blood, glucose coming into the blood, fatty acids coming into the blood. Those are really those nutrient groups. They're all getting absorbed into the bloodstream, and they're all available to cells to be able to make ATP. So you see along the way, there's a number of places that you can make ATP in a cell, and you can use amino acids to do that, or sugars, or fats, fatty acids to be able to do it. This production of energy takes place in one of 2 places, here or here. The cytoplasm of the cell is just the general interior. And this is where the first step of energy production occurs. This is where glycolysis occurs. And you can get some energy from this, but you don't get a whole lot. Really, the place where you get the most amount of energy is the mitochondria. So they blew up the picture of the mitochondria. So the mitochondria looks like this little oblong sort of shape. It has a double layer. So it actually has this double membrane. See, the outside looks like a cover, but the inside is this little ruffly membrane. They call the mitochondria the powerhouse because this is where almost all of your ATP production comes from. So this is truly the energy generator for the cell. So we're really going to focus on glucose. So glucose, C6H12O6, this is glucose. And I wrote this reaction on the bottom of the second page. Glucose, if it can break down completely, this is combustion. Remember combustion, anything plus oxygen making carbon dioxide and water. So we're doing a combustion reaction, much like you could burn wood or you could burn gasoline. But for us, we're going to use enzymes to do this. So we don't have an open flame like you would for fuels if you're trying to generate heat to heat the house. But we can start off with a carbohydrate, add oxygen to it, break the bonds in the carbohydrate all the way down to carbon dioxide and water. So we're taking a big 6 carbon molecule and we're breaking it into smaller and smaller pieces. As you break bonds, you make energy. So breaking chemical bonds is the energy that's going to be able to be used to make ATP. Well, there's a couple of helpers along the way. So they call them like metabolically relevant nucleotides. All they do is they help to move atoms around. Because when you break a bond, you need to have something to be able to pick it up. Because you don't want to have like unpaired electrons, because that would be super reactive and you wouldn't want that. That could cause like random cell damage and all kinds of odd things. So instead, you have these like helper molecules. One of the helper molecules is ATP. So ATP on the left, that's ATP. It's just adenosine with 3 phosphates. That's where its name comes from. And adenosine is a lot like adenine. So adenine is actually one of the nucleotides in DNA. So it's a very similar structure. That extra phosphate is where this energy comes from. So that link is called a high energy bond. Without it, it's only ADP. So adenosine diphosphate just means that it only has 2. You have to take energy in order to add that third one. So it is sort of the energy capture molecule to make ATP. The second one, so this is really the energy capture molecule, so that the energy is not lost. The second one is called NAD and NADH. So this molecule can gain hydrogen. Do you see the hydrogen up here? So it can pick up a hydrogen. The hydrogen comes from glucose. So glucose is C6H12O6. So those 12 hydrogens get plucked off of the glucose. And NAD is responsible for helping to do that. So it's sort of like the receiver of the hydrogen. So as we break glucose down, we're going to pull hydrogens off of the glucose structure. And that is going to be used to generate ATP. Okay? So this is one that's kind of, I just think of it as a short-term hydrogen carrier. So this is a hydrogen carrier in energy production. My spelling is awful looking today. And then my pen acts wonky. So this is a hydrogen carrier. This has a 5-carbon sugar, so it's a pentose. It has that same adenosine. So see how it kind of looks similar to ATP? There's an adenosine, there's a couple of phosphates. But the top part is nicotinamide. That is a vitamin. The B vitamins are really big in metabolism. This is B3. You get this from niacin. Can be made into this. There's also, where's the other one? Oh, I didn't put it in here. Terrible. There's another one that is FAD, and FAD does the same thing. So FAD can pick up 2 hydrogens that get pulled off of glucose and act as a short-term carrier. FAD stands for flavine adenine dinucleotide, and it comes from riboflavin. Riboflavin is vitamin B2. So you need B2 and you need B6 as a normal vitamin supplement. You get these in a lot of grains, a lot of vegetables, so most people are not deficient in these unless they're not eating normal food. But just think about them as being like the hydrogen carriers off of glucose. So in the steps, so that's on, and we'll talk about the last one. We'll talk about coenzyme A in a minute. In the back page, sort of splits the 3 steps of energy production. There's glycolysis, the citric acid cycle, and electron transport. So those are really the 3 major parts. So the first one is glycolysis. So you can just add, I've already got like notes that you can use to kind of follow along through. If you see anything or underline or whatever, just to help. First thing, where does each one happen? So glycolysis happens in the cytoplasm. So glucose comes in. It is actually a process of 10 chemical reactions. So it's not just 1 or 2, there's 10 steps. And the basis is we take this 6-carbon glucose and we're going to split it in half. So that's really the big thing that happens in glycolysis. You take 6 carbons and you're just going to break it into 2 3-carbon molecules. Those 3 carbon molecules are called pyruvate. In the steps, and I kind of changed, I found a newer one. It's just because it's bigger than the one you've got. You don't have to worry about drawing them. You don't have to learn the names of any of the steps that actually occur. But here's the first 5 steps. So if you look at the very top one, can you kind of see how that looks like a little glucose? So it's kind of a ring. They use the little gray dots as being carbons. So this would be like carbon 1, 2, 3, 4, 5, and 6. So in that, you're starting with the glucose. You go through, I told you make ATP in this process, but what happens in this? Seems a little backwards, because what happens to ATP? You're actually breaking it down. So they do call this the energy investing step. You put 2 ATPs in to start this reaction, which seems kind of backwards because supposedly you're making energy, but this actually requires 2 ATP. The ATP is to provide these phosphates. So 1 ATP donates a phosphate, and by doing that, do you see how it sort of shifts the position of the oxygen? Then another phosphate gets added, and that allows you to split it. So that allows that molecule to get split by adding those phosphates to either side. It helps to weaken that center link so that the enzymes can break it. So the thing to remember, this 1 glucose gets broken into 2 3-carbon molecules. You have to start off using 2, but then look what you get. So then in step 6, what do? What did I tell you NAD does? Pulls off hydrogen. So see how NAD now becomes NADH, and you get 2 of them. So that means 2 hydrogens get pulled off. So here, hydrogens get removed. And each one of those 3-carbon molecules produces 2 more ATPs, and you got 2 of them. So how many ATPs total? So you end up with 4. So you start off with the 6-carbon molecule, split it in half, but then each piece will make 2 ATPs. So you end up making 2 ATPs net. So 2 ATPs are used, but 4 ATPs are made. So in that, you still make some energy. So you're going to make a little bit. In glycolysis, 2 ATPs are made per glucose. And we also make some NADs, some NADHs. Okay, so we have some hydrogen carriers that are generated in this step. And we'll talk about what we use those for at the end. Did you see any oxygen used in this step? No. Okay, this is considered anaerobic. You can do this reaction even if there's no oxygen available. So if you exercise, what do you have to do to be able to exercise anaerobically? Okay, breathe or not breathe? So anaerobic versus aerobic. Which one means to breathe? Aerobic. So anaerobic means not breathing. Okay, so have you ever sprinted, okay, or weightlifted to the point, or did the flexed arm hang, like where you did some kind of exercise until you couldn't do it anymore? That's what you have to do in order to kick in this anaerobic energy production. It's made for short term, like high force, short bursts of energy, like a sprint. Okay, so you think like the 50-yard dash where they like had to run like the length of the basketball court as fast as you absolutely could. That's anaerobic production. And then how do you often feel after you do that for a while? Sore, right? So your muscles the next day, you're kind of stiff, you're kind of sore. That's all because of anaerobic energy production. So in this, if we're going to stay aerobic, that means that we have oxygen. So the term aerobic means that there is oxygen present. If you have oxygen, then energy production continues with the next 2 steps. If you don't, then it's anaerobic. So we can make energy anaerobically, but we are going to have lactic acid buildup. So lactic acid, that is the soreness that you feel. And it takes 12 to 24 hours for that lactic acid buildup to actually break down. So you're not sore for days and days and days. It's usually like you get up and you're sore, and then as the day goes on, you feel a little better. You can use that lactic acid. It gets broken back down. You'll use it for energy. And so the more you do this, the better your muscles are at recovery. So that's why people that are big weightlifters that like go and work out and they've got the big bulging muscles, they don't have as much of a sore after exercise effect because they do it constantly. So you can actually build up like tolerance to that sore muscle effect. I had a friend that, I don't know if you've heard of like that P90X, which is like the, you know, the very high force short-term exercise. Like they do they do like workouts for only like 20 minutes. But it's like where they're like throwing those humongous ropes around and walking around with kettlebells and all kinds of, they do the burpees. I'd like fall down the floor and never get back up again. But like she started doing that. Now she's like, she looks like a Jack Russell. Like she's super skinny and like nothing but muscle. But when she first started, like after she did the first exercise, she couldn't roll over in bed. She was like holding on to the bedpost and like pulling herself around. Like her entire body was sore. That was that lactic acid because she did like very short-term high stress exercise. And over time, when she got used to it, her muscles did get stronger. So it wasn't as easy for her to go anaerobic. But the very first week was awfully funny to see her walking around because she was really stiff and really sore. But that's because of the anaerobic. So if you see anaerobic, this means no oxygen. We can still make energy, but we're only going to make about 2 ATPs per glucose. Just as a side note, yeast prefer this. So yeast will actually break down, they use glycolysis, and under low oxygen levels, they'll take the product of glycolysis, which is pyruvate, and they'll convert it to alcohol. And so this is called fermentation. It happens, yeast are very special. They do this because this actually inhibits other microbes from growing around them so they can survive under low alcohol levels that bacteria die off. Other molds and funguses die off, but yeast live there quite happily. So once that was discovered, beer and wine became the big thing. And like they actually found beer and wine products like with the ancient Egyptians. So it's one of those things that was discovered a long, long time ago. So here, this is like the normal. If we start with glucose, we can break it down to pyruvate. So that's the 3-carbon molecule. You make 2 of them. From 1 6-carbon glucose, we end up with 2 pyruvates. We're going to make 2 ATP, but that would be it. And if we stop there, then it's going to get shuttled and become lactic acid. Yeast can do this one, so I added this one in. You don't have to remember the chemical reactions. They just kind of remember the general flow of them. But yeast are the only ones that actually can take pyruvate and convert it to ethanol. And if you look at the two, they're not that much different. But if you have oxygen, then we're going to go forward. Okay, so the reaction continues to the second step. But the second and the third step, these happen in the mitochondria. So pyruvate has to get moved into the little oblong organelle inside of the cell. Along the way, it is going to be transferred to another carrier. And so this carrier is the one that I had on the bottom of the front page. It's called coenzyme A. Coenzyme A, and that's what this is. It is really just the pyruvate carrier. So it helps the pyruvate to move into the mitochondria. Along the way, it takes the 3-carbon pyruvate and it clips the right hand of it off. So see when you look at this 3-carbon molecule up here, see this side? That's carbon dioxide. So it pulls that end carbon on the right off and makes CO2. So carbon dioxide gets clipped off. So what's left is just a 2-carbon group. There's one of the products of the aerobic breakdown of glucose. You make carbon dioxide and water. So there's one of the places that carbon dioxide is made, and it happens as the molecule gets moved into the mitochondria. Notice again, another hydrogen gets pulled off. Okay, so hydrogen gets pulled off as that goes in. All right, so as an interest, if you have lots of energy, so you're a couch potato, so your muscles are just hanging out, but there's plenty of glucose, you will actually start glycolysis because it doesn't require oxygen. But if you make tons and tons of pyruvate, it has to go somewhere. So the cell doesn't want that to happen. The cell is kind of like in a production assembly line, and it's only going to make energy if there's energy that's needed, because it's sort of a waste of time to make energy if you don't need it. So early in glycolysis, out of the 10 reactions, way up in step 3, an enzyme's there that is actually inhibited by pyruvate. So if you have plenty of pyruvate, this enzyme is inhibited. Remember, we talked about an inhibitor. So an inhibitor binds to the enzyme, and that's going to block it from being able to continue. So it works pretty well. If you start using pyruvate, if you start working out, so your muscles are now needing energy, then pyruvate levels go down. Pyruvate comes off of this enzyme, and the enzyme begins making more, sending more glycolysis products along. Interestingly, fructose is not regulated like this, okay? So here's our regulation step. So I kind of like their little flow through. So they're saying, okay, like here's your production line. So at the top, if glucose comes in and there's plenty of the little red triangles, plenty of pyruvate, plenty of glycolysis products, then that will have a negative effect up here. What about fructose? When fructose comes into glycolysis, it enters glycolysis at step 5. So that means it enters glycolysis past the regulation step. So if lots of fructose comes into the cell, it'll automatically get converted to pyruvate. And you can have large amounts of pyruvate build up in the cell, and the cell can't handle that. So what the cell does is it converts it to fat. It'll actually take pyruvate and build triglycerides. It'll build fat, and where does it send that fat? To your adipose tissue, right? To subcutaneous tissue, to deep organ tissue. So excess pyruvate ends up becoming triglycerides. And so if you've been to the doctor and they say, oh, your triglyceride levels are high, that doesn't necessarily mean that you've been eating a lot of fat. It could also mean you've been eating a lot of fructose. So we kind of talked about fructose earlier. Where do you get fructose? Where is that now very common? Well, that's actually rarely the case. It's rarely the reason that you're going to end up with a lot of triglycerides. Because when you eat fruit, you have a lot of soluble fiber. Remember that helps to slow absorption of those sugars. Where do you get a lot of fructose? In that high fructose corn syrup. So remember, high fructose corn syrup, super sweet, okay? It's actually 150 times sweeter than the average sucrose is. And so that sweet creates the demand or the drive. And so when you start to drink the sugary stuff or eating the sugary snacks, how it starts to create the craving. Well, in that, you're ingesting fructose, which goes into glycolysis at this level. And so people are like, I have a low-fat diet. I don't understand why I keep gaining weight. Well, any carbohydrates that are not utilized, not necessary. The cell doesn't hold on to these pyruvate molecules. It just converts it to fat and sends it off to adipose tissues for storage. So that's the big challenge, is if you're eating all glucose, at least that would get regulated. It wouldn't get broken down. But if you're eating fructose, it actually ends up getting shuttled through digestion and causes a lot more problems. Going on to the next one. Second step. So now we're in the mitochondria. Okay? So in the mitochondria, we need to take and break the 2 carbon molecule down that came in with the acetyl group, the coenzyme A. We need to break it down completely, and that happens in the process called the citric acid cycle. So there's a couple of names they came up with the citric acid cycle. I'm not really sure why, but if you see citric acid cycle, the citric acid cycle, like the Ohio State, if you see TCA, means the same thing. If you see Krebs cycle, same thing. That's all exactly the same cycle. I know. Because Dr. Krebs is the one that found it. So remember, everything used to always be named after the scientist. Well, they've kind of moved away from that. And so citric acid is involved in this reaction. So that's how its name came up. But it's a cycle, which means it's a circular series of reactions. So it doesn't go from here to here. You come in and there's 8 reactions that take place in sort of a circle. So you end up reforming, being able to reform the initial reactant and continue this. So it's going to take 2 turns of the cycle. So each acetyl-CoA, each CoA brings in that 2 carbon molecule. And since you made 2 of them in glycolysis, you're going to have to do this twice. So in this, you're going to make, and we'll follow it along. So there's 8 steps total. You don't have to remember that, but there's 8 steps. Come here. So here we're going to start all the way at the top. So all the way at the top, see the purple. So that purple block, that's the 2 carbon group that was brought in by the coenzyme A. So it comes in and it gets added. Now we've got these 2 carbon with 2 oxygen groups on there. So we have really all that happens is you start to rearrange bonds. So from step 1 to step 2, it's really just adding that 2 carbon group. Step 2 to step 3, chemical bonds get rearranged. So if you sort of look at them to figure out, okay, where's this change that's actually occurring? So notice here, do you see this alcohol group gets shifted from carbon 2 down to carbon 3? That's really all that happens. Okay, but now from this one down to the next, look what gets bumped off. So we're going to pull off a carbon dioxide. So that carbon dioxide, that COO negative, gets pulled off and more hydrogens. So we're going to pull off carbon dioxide. We're going to pull off more of the hydrogens that originated with the glucose. Then we're going to do another bond rearrangement. And in doing so, we're going to allow another carbon dioxide to get pulled off and another hydrogen to be removed. Continuing on along, the only place you make energy in the citric acid cycle directly is right here. So you only make 1 ATP. You made 2 in glucose, in glycolysis. But here you only make 1, okay? Coming up, remember I told you there was that NAD to NADH? Well, here's an FAD, that flavine adenine dinucleotide adds this, pulls off 2 hydrogens. Then we get just a couple more bond rearrangements. All that's happening kind of in the last 2 steps is really just to reform the molecule so that another 2 carbon group can come in. So in this process, 2 carbon dioxides. How many NADHs? Count those. They're the orange blocks. So you get 3 NADHs. And then how many FADH2s? They're the green. Just 1. And then we get 1 ATP. So that's in this one turn. So if you sort of follow along, you see 3 orange boxes. Those are the NADHs made. 1 FADH2, that's the green box. The blue ones, those are the CO2s. There's 2 of those bumped off down near the bottom. And then that 1 ATP in the bottom left-hand corner. 2 turns. So all of that twice as much is made. So it's got to go around 1 time, got to go around a second time to account for 1 glucose. So we get 2 ATPs, so about the same as glycolysis.
Should it be, it says 6 on our sheet, but it should be 4 per CO2.
Right, but remember, where did the first CO2 come off? So remember up here, we took pyruvate. In order for it to go into the mitochondria, carbon dioxide came off,
Yeah.
right? So we pulled one off to go from a 3-carbon group to a 2-carbon group, and then that purple block is the 2-carbon group that's brought in. So it's kind of in the in-between. It's not really in glycolysis. I would say it's closer to belonging to the citric acid cycle if you were going to connect it to something. So if you have C6H12O6, all those carbons get pulled off as carbon dioxide in this cycle. So this is where your carbon dioxide gets made. Did you see any water though in this? No. So where's the water? So the water happens in the last step. So in this happens in the mitochondria. You're systematically breaking down the 3-carbon group, pulling off carbon dioxides, pulling off hydrogens. 2 ATPs get made directly. This is sort of the summary. And again, you don't have to remember the exact number. We'll talk about summary numbers at the very end. But just remember, most of your or all of your CO2 really takes place in this step. This is where your carbon dioxide is generated. Because this is the last step. So we made all these NADHs, we made all these FADH2s in the previous steps. Here's where they come into play. So NADH and FADH2, they transfer these hydrogens to enzymes. There's enzymes in the inside ruffled part of the mitochondria, enzymes that'll take a hydrogen and split it into a hydrogen ion and an electron. So they call this, one of the terms is called the electron transport chain, because that's what it does. If you follow along the little purple proteins, so these are just enzymes. These are proteins, integral proteins in the membrane. They transfer the electrons from complex 1 past Q to complex 3 and then over to complex 4. So it really just passes it along. And that movement, though, powers the pumping of hydrogen ions out of the membrane. So this movement of electrons allows these hydrogen ions to get put from the inside, the very inside of the mitochondria, into that outer membrane space. So what do you start to generate? So we're going to kind of like use this as a pump. We're going to transfer electrons, and in doing that, we're going to shoot hydrogen ions out of the mitochondria. So you end up with high concentration of hydrogen ions outside of that membrane, low concentration of hydrogen ions inside. So is that the normal way of things, to have high concentration, low concentrations? What do they want to do? They want to go back to being equal. So that's like the natural drive, is for things to always go from high to low. So this is called active transport, because it is specifically moving things from where they're at low concentration to where they're at high concentration. So the little picture I drew at the bottom, I'm like, so now you get lots of hydrogen ions in that space outside of the ruffle, but inside of the outer membrane. So in that little space in the mitochondria, now you have lots of hydrogen ions that build up. And inside, you don't have very much at all. So we have high concentration and low concentration. That allows the last step. And the last step is where this ATP generator kicks in. So there is a last enzyme all the way at the end of this that is called ATP synthase. So it synthesizes ATP. The only thing that's allowed to pass through is hydrogen ions. So they go from high concentration, they come back into the inner mitochondrial membrane. So these hydrogen ions, these little H pluses, come shooting back in. As they do, that movement, that flow from high concentration to low concentration has the energy to be able to build ATP. So movement of those hydrogens back into the mitochondria allows that ATP generation. And then the last thing, so those electrons that get passed, these hydrogen ions that come back in, they combine with oxygen and form water. So water is actually made in this last step. Carbon dioxide is made in the citric acid cycle. That's in step 2. Oxygen is here. So oxygen is necessary in order for complete combustion of glucose to occur. If you don't have that oxygen, you can't do this step. But if you don't have that oxygen, then the second step begins to stop because there'll be so much NADH and FADH2 hanging around that you can't go any further. You only have a certain number of these hydrogen carriers that are available inside of the mitochondria. So if they're all full and you don't have any oxygen, you can't run this step, so you can't run the second step either. So they call the citric acid cycle and electron transport, they're both considered aerobic. You have to have oxygen in order to do these two. In order for the citric acid cycle and electron transport to occur, you've got to have oxygen. So how much difference does this energy production occur? So in glycolysis, we said that you make 2 ATPs. So there's 2. There's 2 NADHs that are made in glycolysis. Every NADH generates 2.5 ATPs when it goes through the electron transport. So if that NADH can go through step 3, it'll make 2.5. So how many ATPs there? 5. Okay, so that step going down into the mitochondria when pyruvate bumps off a carbon dioxide, making the little 2-carbon carrier, that'll also make 2 NADHs. So there's hydrogens pulled off there. So that'll give you 5. Citric acid cycle is where you get the most NADH and FADH2 molecules. So you get 2 ATPs, which are GTPs. So that would be, they're equal. A GTP and an ATP are equivalent. You get 6 NADHs. So how many ATPs is that? 15. So you make 15 ATPs. FADH2, you make 2 of them because that cycle goes around 2 turns, 1 in each turn. So you only get 1.5. So if you jump back 1, do you see that FADH2 comes in later? You don't get as much ATP as you do from the NADH. So how many ATPs total? 3. So add those all up. And you get? So 1 molecule of glucose can make 32 ATPs. So do you see why I said with glycolysis you don't get a whole lot of ATPs? If you can run steps 1, 2, and 3 from 1 molecule of glucose, you can get 32 ATPs. And as long as you have oxygen, you can do this aerobically. So this is why like moderate exercise, so like if you go out and you're walking, you can do this as long as you don't run out of nutrients, as long as you have oxygen, you can run this and run this and run this just consistently. So you can do that exercise for hours, okay? People that are like runners, they'll train themselves to improve their lung capacity so they'll actually not run out of oxygen. They make sure that their diet, that they're inputting the nutrients so that they can break it down and then go out and run for 4 and 5 hours. Ridiculous. There are some people that are, have you heard of like these distance runners that will run for like 10 hours straight? I just don't get it. But it's like they have to be just as efficient as possible. So that means that their skeletal muscle is capable of kicking in aerobic energy and powering their muscle cells for 10 hours straight. So they're like, yeah, that's like a huge, it's just crazy. Like their muscle cells, that's just crazy. Okay, so 32 ATPs. So what happens if you use fats for energy? So remember in this, so we can use fatty acids. Notice that if we use fatty acids, we really don't go through, this is glycolysis. We really don't use glycolysis. If we're going to break down fats, you actually take the fatty acids and you clip off 2 carbons from those fats. Those are long chains of carbon. So you just clip off 2 units at a time, and those 2 units get shuttled in as acetyl groups. Within the TCA cycle, those little 2 carbon groups can get put in. So here's this one. So this is one fatty acid chain. And so in the processing, you again make an NADH, you make an FADH2, and you can clip off 2 carbon units. So you clip off 2, clip off 2, clip off 2, and feed that in. The problem comes into play is some of these byproducts. They call this process beta oxidation, the breakdown, and you make a ketone. So one of the byproducts are keto acids. Keto acids are formed in the utilization of fats for energy. Normally, small amounts can get filtered out by the kidneys. But this is one of the reasons that people that are on the keto diets, that they tell them to make sure that they drink large amounts of water. You really should drink a gallon of water a day, which seems like a huge 124 ounces of water a day. That's a lot of water. Okay? But that is why, is because those acids end up getting filtered out by your kidneys. And so if you're not drinking a lot of water, then that means it's in a higher concentration, and that's more stressful on the kidneys. So patients that have diabetes and don't take insulin, they also make those keto acids. And remember, we were talking about acidosis. Keto acids can actually lead to a change in the blood pH. So it's not as much of a problem for somebody that is doing like a low carb type of diet, because they're also eating enough carbohydrates and they're not suffering from hyperglycemia. Right? So if they're on the low carb diet, you're not eating carbohydrates, so you would never have really high blood sugar, which is also the problem that the diabetic has. So they sort of have the double problem. So they will have high fat metabolism. They will make a lot of keto acids, and they're going to have very high blood sugar. So the combination of those ends up leading to that ketoacidosis and can lead to the acidosis of the blood, can lead to loss like depression of nervous system function, can put the person in a coma. And so the balancing of the two is the challenge. So last one, where's the last one? The only other one is, what if you use proteins? And if you're like, well, I won't eat any fat and I won't eat any carbohydrates, I'll just eat proteins. You can make energy from proteins. All you have to do is use your amino acids. Why is it not? It's like, no, I don't want to. So in this case, remember that all of your amino acids have this. And that's not found in glucose, and that's not found in fats. So this is nitrogen. And this is where all of your nitrogen waste comes in. So the nitrogen waste is called urea. And so if you're burning a lot of proteins, burning a lot of amino acids, in order to be able to use amino acids for energy, you have to take that nitrogen off. So that means that you're going to make more nitrogen waste. So again, what organ does that stress out? Kidneys. Your kidneys again, okay? So having high protein in your diet, again, increasing water intake will help to dilute out the amount of urea that's produced, because it'll be much higher than somebody that, say, eats like mostly like the low carb like the low fat, low protein type of diet. They're going to have less of that nitrogen waste. So having high amounts of protein in the diet leads to more nitrogen waste. And again, this is the sort of a kidney stress. So you can use all three of those. But usually, what do you want to use your amino acids for? Yeah, to build muscle, to be able to build proteins that the cell needs. So optimally, you would do this last. You would rather burn carbohydrates than burn fats as a supplement, and burn amino acids absolutely last, because you want to use those to build muscle, to build enzymes, to build collagen and other things the cell needs. All right, so on this is kind of like just the general summary for them. For each of the steps is really the best way to go about. I don't expect you to like be able to draw any pathways or like name any of the enzymes, but go through, and if you have just a general outline of what happens in each step, then you should be fine with this. If you did not get your 5th exam, I have it.